Common protocols in molecular biology. Chapter 9. Electrophoresis. Principle of electrophoresis. Electrophoresis is the process of moving charged molecules in solution by applying an electric field across the mixture. Because molecules in an electric field move with a speed dependent on their charge, shape, and size, electrophoresis has been extensively developed for molecular separations. As an analytical tool, electrophoresis is simple and relatively rapid. It is used chiefly for analysis and purification of very large molecules such as proteins and nucleic acid, but can also be applied to simpler charged molecules including charged sugars, amino acids, peptides, nucleotides, and simple ions. Highly sensitive detection methods have been developed to monitor and analyze electrophoretic separations. Electrophoresis of macromolecules is normally carried out by applying a thin layer of a sample to a solution stabilized by a porous matrix. Under the influence of an applied voltage, different species of molecules in the sample move through the matrix at different velocities. At the end of the separation, the different species are detected as bands at different positions in the matrix. A matrix is required because the electric current passing through the electrophoresis solution generates heat, which causes diffusion and convective mixing of the bands in the absence of the stabilizing medium. The matrix can be composed of a number of different materials, including paper, cellulose acetate, or gels made of polyacrylamide, agarose, or starch. In acrylamide and agarose gel, the matrix also acts as a size-selective sieve in the separation. At the end of the run, the separated molecules can be detected in position in the gel by staining or autoradiography, quantified by scanning with a densitometer in the gel can be dried for permanent storage. Polyacrylamide and agarose gels are the most common stabilizing media used in research laboratories. The gel are usually formed as cylinders in tubes or as thin, flat slabs or sheets. Polyacrylamide is the most common matrix for separating proteins. Nucleic acid are separated on either polyacrylamide or agarose gels depending on the size of molecules to be analyzed. Figure 1. Basing arrangement for electrophoresis. Agarose electrophoresis. Introduction. Agarose, an extract of seaweed, is a polymer of galactose. The polymer is 1,3 linked between brackets beta, D-galactopyranose, and 1,4 linked 3,6 anhydro between brackets alpha, I-galactopyranose. The primary applications are electrophoresis of nucleic acids, electrophoresis of very large proteins, and immunoelectrophoresis. After a PCR assay, between brackets or during an assay with some cycler systems, several crucial questions need to be answered in order to determine the success of the assay. How do we know if the PCR has worked? How should we interpret the results? What do we need to do with the PCR applicants? DNA is negatively charged and these molecules will migrate to the positive electrode between brackets cathode when placed in an electrical field. The ratio of a charge to mass in DNA is uniform and therefore all linear DNA fragments will possess the same electrophoretic properties when subjected to electrical current in a medium with no resistance. Nonlinear DNA molecules, like covalently closed circular molecules, will have electrophoretic migration patterns that are not directly proportional to mass. Due to differences in density and the conformation of these coiled DNA species, agarose is an easy and cost-effective medium that, in gel form, provides the viscosity that will allow the DNA molecule to separate in an electrical field. Based on size and conformation, the size of the matrices in agarose gels is determined by the percentage of agarose in the gel and may be manipulated to allow the optimal electrophoretic efficiency depending on the size ranges of the expected applicants of the particular PCR analysis. Electroendosmosis between brackets MR or EEO. MR is a measure of the amount of electroendosmosis that occurs during electrophoresis with a particular grade of agarose. Electroendosmosis is a mass movement of water towards the cathode against the movement of the macromolecules, which is usually towards the anode. High MR means high electroendosmosis. The mass flow of water toward the cathode is caused by fixed negative charges in the agarose gel between brackets sulfate and the carboxyl groups on the agarose. Depending on the application, electroendosmosis causes the loss of resolution or it can enable certain kinds of separations to occur. For instance, during counter-immunoelectrophoresis. Percentages of agarose for DNA separation. 0.3% of agarose, 1,000 to 30,000 size of linear DNA. 0.7% of agarose, 800 to 12,000 size of linear DNA. 1.0% of agarose, 500 to 10,000 size of linear DNA. 1.2% of agarose, 400 to 7,000 size of linear DNA. 1.3% of agarose, 300 to 3,000 size of linear DNA. 1.4% of agarose, 250 to 3,000 size linear of DNA. 1.5% of agarose, 200 to 3,000 size of linear DNA. 2.0% of agarose, 50 to 2,000 size linear of DNA. Buffers. Two types of buffers are generally required. One, loading buffer. This is the buffer to be added to a sample of the PCR reaction that will be electrophoresed. The buffer contains a colorant like bromophenol blue that will allow you to visualize the sample when loading the gel and to track the migrating material in the gel during electrophoresis. 2. Electrophoresis buffer. This is the buffer covering the gel, cathode and anode, and contains appropriate ionic salts that will dissociate and travel across the agarose matrix with the movement of electrons from the cathode to the anode. Electrophoretic conditions. The conditions of the electrophoresis depend on 1. The strength of current between brackets amperes. 2. The power applied between brackets watt. 3. The potential difference between bracket volt. These are dependent on the specific applications, size of the gel, and the percentage of agarose in the gel, between brackets, density of the matrix. Most commonly, the criteria of 5 volts per centimeter of gel is applied and results in a total voltage of 60 to 100 for most gels. In general, higher voltages results in faster separations, but lower voltages permit more accurate separations and resolution of different DNA species. Inspection of gels. After electrophoresis and staining of the gel, inspection on a UV translaminator will allow one to determine whether there is an amplification product, 2. The quality of the product, 3. The specific size of the product. The latter is achieved by the routine inclusion of a DNA standard that typically contains a range of fragments of known size. It is the most important, however, that the presence of an applicant after PCR cannot confirm the specificity of the reaction, i.e., the genetic makeup of the product. At the most, the presence of an applicant of exactly the expected size is an indication that the PCR has targeted the specific templates you have intended for it. Confirmation of the integrity and the identity of the applicant may be achieved by a variety of additional methods, including nested PCR, hybridization, restriction length, polymorphism between brackets, RFLP, and sequence analysis. Visualizing the DNA. After the electrophoresis has been completed, there are different methods that may be used to make the separated DNA species in the gel visible to the human eye. The most common of these is to stain the gel with ethidium bromide and then visualize the gel on a UV translaminator. The ethidium bromide intercalates with the DNA molecules, and this agent will transmit UV lights as long-wave fluorescence that is easy to see and photograph. Many researchers opt to include ethidium bromide in the loading buffer and or in the electrophoresis buffer, saving time on the staining step after electrophoresis. This is perfectly acceptable, but because ethidium bromide is an intercalating agent, 2 important issues should be kept in mind. Firstly, when ethidium bromide intercalates and associates with the base pair in the 2 DNA strands, these helical molecules are rendered stiff and rigid, affecting their migration through the agarose matrix. This effect is not serious, but staining should rather be done after electrophoresis in cases where the accuracy of the size measurements of DNA amplicants is a premise. Secondly, ethidium bromide is a mutation that intercalated substance will also cause breaks and nicks in DNA strand when exposed to UV, between brackets including sunlight. The compound is therefore hazardous and should be handled with extreme care. Other agents for staining include silver, which is very effective but an expensive alternative. Basic protocol for agarose gel electrophoresis. Materials: 1 horizontal gel electrophoresis apparatus, 2 microwave oven or heating plate, 3 power supply, 4 UV translaminator, 5 electrophoresis buffer between brackets 1x TAEE, 6 ethidium bromide between brackets ATBR. Add 0.2 gram of ethidium bromide to 20 milliliter of H2O. Mix well. Store at 4 degrees Celsius in dark between brackets brown or foil covered bottle. 7 agarose, 8 gel loading buffer to make 10 milliliter final concentration. Glycerol 5 milliliter 50% final concentration. 4x TAE buffer 250 microliter 1x final concentration. Bromophenol blue saturated 1 milliliter 1x final concentration. Xylene cyanol 10% suspension 1 milliliter 1% final concentration. H2O 2.75 milliliter. 9 DNA molecular weight marker 10 TAE buffer 40x to make 1 liter 3 base between brackets 1.6 m 193.6 gram. Sodium acetate 3H2O between brackets 0.8 m 108.9 gram. ADTA sodium 2H2O between brackets 40 mM 15.2 gram. pH 2 7.2 with acetic acid H2O to make 1 liter. Methods: 1. Prepare and educate volume of 1x TAE buffer to fill the electrophoresis tank and prepare the gel. To facilitate visualization of DNA fragments during the run, ethidium bromide solution can be added to the electrophoresis buffer to a final concentration of 0.5 microgram per milliliter. 2. The amount of agarose that is used depends on the size of the tray and the concentration required. For general purposes of 1% gel is used. Gels are typically between 0.5 and 1 cm thick. 3. Prepare a solution of molten agarose as required by adding the 1x TAE buffer to the appropriate amount of agarose powder in a suitable EM flask. Bring to boil in the microwave oven. The agarose must be completely dissolved prior to pouring. Adjust volume with distilled water to compensate for any loss. 4. Seal the end of the plastic tray with masking tape and pour the gel after the agarose has been allowed to cool down to about 55 degrees Celsius. This prevents warping of the gel apparatus. Insert the comb and make sure that there is no bubbles trapped underneath the comb and that all bubbles on the surface of the agarose are removed before the gel sets. 5. After the gel has set, remove the tape from the casting tray and withdraw the gel comb, taking care not to tear the sample wells. 6. Place the gel casting tray containing the set gel in the electrophoresis tank. Add sufficient 1x TAE buffer to cover the gel to a depth of 1 milliliter between brackets or until the wells are just submerged. Make sure no air pockets are trapped within the wells. 7. Apply 5 microliter of the gel loading buffer to each microliter sample and add samples to the individual wells. Take care not to overload and be sure to include appropriate DNA molecular weight markers. 8. Cover with the safety cover and run gel to 110 milliampere between brackets 7 times 10 centimeter tray or 200 milliampere between brackets 15 times 10 centimeter tray. Typically 1 to 10 volt per centimeter of gel. When the bromophenol blue marker is about 2/3 from the top, the gel can be stained with ethidium bromide between brackets ATBR. 9. Turn off the power supply when the bromophenol blue has migrated a distance judged sufficient for separation of the DNA fragments. 10. The DNA can be visualized on a UV translaminator and photographed. 11. Gels have been run in the absence of ethidium bromide can be stained by covering the gel in a dilute solution of ethidium bromide between brackets 0.5 microgram per milliliter in water and gently agitating for 10 to 30 minutes if necessary. Gels can be destained by shaking in water for an additional 30 minutes. This serves to remove excess ETBR that cause background fluorescence and make visualization of small quantities of DNA difficult. SDS polyacrylamide gel electrophoresis of proteins. SDS page is the most widely used method for qualitatively analyzing protein mixture. It is particularly useful for monitoring protein purification, and because the method is based on the separation of proteins according to size, the method can be also used to determine the relative molecular mass of proteins. Formation of polyacrylamide gels. Cross-linked polyacrylamide gels are formed from the polymerization of acrylamide monomer in the presence of a smaller amounts of N, methylene BIS acrylamide between brackets, normally referred to as BIS acrylamide. Note that BIS acrylamide is essentially two acrylamide molecules linked by a methylene group and is used as a cross-linking agent. Acrylamide monomer is polymerized in a head-to-tail fashion into long chains, and occasionally a BIS acrylamide molecule is built into the growing chain, thus introducing a second site for a chain extension. Proceeding in this way, a cross-linked matrix for fairly well-defined structure is formed. The polymerization of acrylamide is an example of free radical catalysis and is initiated by the addition of ammonium persulfate and the base N, tetramethylene diamine between brackets TAMED. TEMED catalyzes the decomposition of the persulfate ion to give a free radical. The use of stacking gels. For both SDS and buffer gels, sample may be applied directly to the top of the gel in which protein separation is to occur, between brackets, the separation gel. However, in these cases, the sharpness of the protein bands produced in the gel is limited by the size between brackets volume of the sample applied to the gel. Basically, the separated bands will be as broad between brackets or border owing to diffusion as the sample band applied to the gel. For some work, this may be acceptable, but most workers require better resolution than this. This can be achieved by polymerizing a short stacking gel on top of the separating gel. The purpose of this stacking gel is to concentrate the protein sample into a sharp band before it enters the main separating gel, thus giving sharper protein bands in the separating gel. This modification allows relatively large sample volumes to be applied to the gel without any loss of resolution. The stacking gel has a very large pore size between brackets 4% acrylamide, which allows the protein to move freely and concentrate or stack under the effect of the electric field. Sample concentration is produced by isotachophoresis of the sample in the stacking gel. The band sharpening effect between brackets isotachophoresis relies on the fact that the negatively charged glycinate ions between brackets in the reservoir buffer have a lower electrophoretic mobility than the protein SDS complex, which in turn have lower mobility than the chlorine ion if they are in a region of higher field strength. Field strength is inversely proportional to conductivity, which is proportional to concentration. The result is that the three species of interest adjust their concentration so that concentration of chlorine ion greater than protein SSD greater than glycinate. There are only a small quantity of protein SDS complex, so they concentrate in a very tight band between the glycinate and the chlorine ion boundaries. Once the glycinate reaches the separating gels, it becomes more fully ionized in the higher pH environment and its mobility increases. Between brackets, the pH of the stocking gel is 6.8 and that of the separating gel is 8.8. Thus, the interface between glycinate and the chlorine ions leaves behind the protein SDS complex, which are left to electrophorese at their own rates. Figure 2. Sodium dodecyl sulfate based polyacrylamide gel electrophoresis between brackets SDS page. A. Polypeptides applied to a gel slot accumulate to a small band during their migration through the electric field in the stacking gel forward by the electric field. B. In the presence of SDS, polypeptides are separated according to their molecular masses as the anionic detergent SDS binds to polypeptide chains, giving them an overall negative charge. SDS page. Sample to be run on SDS page are first boiled for 5 minutes in a sample buffer containing beta mercaptoethanol and SDS. The mercaptoethanol reduce any disulfide bridges present that are holding together the protein tertiary structure. SDS is an anionic detergent and binds strongly to and denature the protein. Each protein in the mixture is therefore fully denatured by the treatment and opens up into a rod-shaped structure with a series of negatively charged SDS molecules along the polypeptide chain. On average, one SDS molecule binds for every 2 amino acid residues. The original native charge on the molecule is therefore completely swamped by the SDS molecules. The sample buffer also contains an ionizable tracking dye, usually bromophenyl blue, that allows the electrophoretic run to be monitored, and the sucrose of glycerol, which gives the sample solution density, thus allowing the sample to settle easily through the electrophoresis buffer to the bottom when injected into the loading well. When the main separating gel has been poured between the glass plates and allowed to set, a shorter sticking gel is poured on the top of the separating gel and it's into the gel that the wells are formed and the protein loaded. Once all samples are loaded, a current is passed through the gel. Once the protein sample have passed through the gel, stacking gel and have entered the separating gel, the negatively charged protein SDS complex continue to move toward the anode, and because they have the same charge per unit length, they travel into the separating gel under the applied electric field with the same mobility. However, as they pass through the separating gel, the proteins separate owing to the molecular sieving properties of the gel. Quite simply, the smaller the protein, the more easily it can pass through the pores of the gel, whereas larger proteins are successively retarded by fractional resistance owing the sieving effect of the gel. Being a small molecule, the bromophenyl blue dye is totally unretarded and therefore indicates the electrophoresis front. When the dye reaches the bottom of the gel, the current is turned off and the gel is removed from between the glass plates, shaken in an appropriate stain solution between brackets, usually C brilliant blue, for a few hours and then washed in distant solution overnight. The distant solution removes unbound background dye from the gel, leaving stained protein visible as blue bands on a clear background. A typically large format gel would take about 1 hour to prepare and set, 3 hours to run at 30 milliampere, and have a staining time of 2 to 3 hours with an overnight stain. Mini gels run at 200 volt constant voltage can run in about 40 minutes and require only 1 hour staining. Most bands can be seen within 1 hour of the staining. Materials and equipment. 1 vertical slab gel unit with casting stand, 2 1.5 or 0.75 milliliter thick combs and speakers, 3 glasses plates, 4 50 milliliter and 125 milliliter side arm flask, 5 transfer pipette, 6 pipetter, between brackets 1 to 20 microliter with long narrow gel loading pipette tips, 7 water aspirator or vacuum pump with trap, 8 boiling water bath or 100 degrees Celsius temperature block, 9 power supply between brackets 300 volt, 10 magnetic stirrer, 11 pipette graduated cylinder, 12 molecular weights protein strands. Method: 1. Prepare the separating gel. Assemble the SE600 vertical slab gel unit in the dual gel casting stand. Use 1.5 milliliter or 0.7 milliliter speakers. 2. In a 125 milliliter side arm vacuum flask, mix either 40 milliliter between brackets 0.75 milliliter or 80 milliliter between brackets 1.5 millimeter of resolving gel solution, leaving out the ammonium persulfate and the TEMED. 3. Stopper the flask and apply a water vacuum for several minutes to deaerate the solution while swirling the flask. 4. Add the TEMED and ammonium persulfate and gently swirl the flask to mix, being careful not to generate bubbles. 5. Pipette to solution down the speaker into each sandwich to a level about 4 cm from the top. A 25 milliliter pipette works well for this step. 6. Fill a transfer pipette or syringe with water saturated and butanol between brackets or water. Position the pipette or needle at about a 40 degrees Celsius angle with the point at the top of the acrylamide next to a speaker. Gently apply approximately 0.3 milliliter of N-butanol. Repeat on the other side of the slab next to the other speaker. The N-butanol layer evenly across the entire surface. After a minute or two, repeat this process to overlay the second slab. A very sharp liquid gel interface will be visible when the gel has polymerized. This should be visible within 10 to 20 minutes. The gel should be fully polymerized in about 1 hour. 7. After polymerization, tilt the casting stand to pour off the overlay and rinse the surface of the gels twice with resolving gel overlay. Gels may be stored at this point. The stacking gel is cast later. Remove the N-butanol and add approximately 10 milliliter of one resolving gel overlay solution to the top of each sandwich. Seal with plastic wrap and store flat at 4 degrees Celsius, or store gels submerged flat in 1x resolving gel overlay at 4 degrees Celsius for up to 1 WK. 8. Add approximately 1 milliliter of resolving gel overlay to each gel and allow the gels to set while preparing the stacking gel. Prepare the stacking gel. 9. In a 50 milliliter side arm vacuum flask, mix 10 milliliter between brackets for 0.75 millimeter thick gel or 20 milliliter between brackets for 1.5 milliliter thick gel of a stacking gel solution, leaving out the ammonium persulfate and the TEMED. 10. Deaerate as in step 3. 11. Add the ammonium persulfate and the TMED. Gently swirl the flask to mix. 12. Pour off resolving gel overlay from the gel. Remove all liquid before proceeding. 13. Fill each sandwich with a stacking gel solution and insert a comb into each sandwich, taking care not to trap any bubble below the teeth of the comb. 14. Allow the gel to set for at least 30 minutes. A very sharp interface will be visible when the gel has polymerized. This should be visible within 10 to 20 minutes. The gel should be fully polymerized after 1 hour. In general, stacking gels should be cast just before use. The complete gel can be stored overnight at 4 degrees Celsius, however, with little effect and resolution if covered and the comb left in place. Prepare the sample. 15. Combine equal volumes of protein sample and the 2 treatments buffer in a tube and place the tube in a boiling water bath for 90 seconds. If using dry sample, Add equal volumes of water and two treatment buffer and heat in a boiling water bath for 90 seconds. This will be diluted by the 2x sample buffer to give 5 to 10 microgram per microliter for complex mixture between brackets E.G. cell lysates. 50 microgram of proteins between brackets 5 to 10 microliter of treated sample. Perlin is recommended for highly purified protein. 0.5 to 5 microgram per lane is usually adequate. Silver staining requires 10 to 100 fold less protein per lane. 16. Place sample briefly on ice until ready for use. The treated sample can be stored at 20 degrees Celsius for 6 months before future runs. Load the gels. 17. Slowly remove the cum from the gels, raising the cum up to avoid disturbing the well's dividers. 18. Rinse each well with tank buffer. Invert the casting stand to drain the wells and return the stand to an upright position. 19. Fill each well with tank buffer. 20. Using a pipetter with a long thin tip between brackets or a Hamilton syringe, gently load 5 to 10 microliter of sample beneath the buffer in each well. Load every well with the same volume of sample. If the well is not needed, load with 1x sample buffer containing standard protein or no sample. 21. If protein molecular weight strands are used, load 1 or 2 wells with 5 to 10 microliter of the strand mixture. If the gel is to be stained with coma C blue, the volume should contain 0.2 to 1 microgram of each standard component. If the gel is to be silver stained, use 10 to 50 ng of each component. Round the gel. 22. Fill the lower buffer chamber with electrophoresis buffer. Install the sealing gasket and the upper buffer chamber and put it into place on the gel sandwiches. Put the upper buffer chamber in place on the heat exchanger in the lower buffer chamber. 23. Adjust the height of the tank buffer in the lower buffer chamber until the sandwich are fully immersed in buffer. If bubbles are trapped under the ends of the sandwich, coax them away with a pipette. 24. Add a spin bar of the lower buffer chamber and center the chamber on a magnetic stirrer. When the lower buffer is stirred, the temperature of the buffer remains uniform. This is important because uneven heating distorts the band pattern of the gel and leads to smelling. 25. Carefully fill the upper buffer chamber with tank buffer. Do not pour buffer into the sample wells because it will wash the sample out. 26. Put the lid on the gel unit and connect it to the power supply. The cassette between brackets black lid is connected to the upper buffer chamber. 27. Turn on the power supply and adjust the voltage. 28. Adjust the current to 30 mA per 1.5 mm thick gel and 15 mA per 0.75 mm thick gel. Start the electrophoresis. The voltage should start at about 70 to 80 volt but will increase during the run. Keep a record of the voltage and the current readings so the future runs can be compared and current leaks or incorrectly made buffers can be detected. 29. When the tracking die reaches the bottom of the gel, turn the power supply off and disconnect the power cables. Desensible the gel sandwich. 30. Remove the buffer and desensible sandwiches. Carefully lift the gel into a tray of staining solution or fixative. Procedures of degazing acrylamide gel solutions. Degaze acrylamide solution in a sidearm vacuum flask with a cork that is wider than the flask opening for 15 minutes with gentle stirring. Use at least a bench vacuum to degaze. A water aspirator on the sink is not strong enough. When the solution bubbles up and threatens to overflow into the sidearm, release the vacuum by quickly removing the cork from the top of the flask. Then replace the cork, swirl the solution, and continue the procedure. The solution will bubble up 4 or 5 times and then most of the air will be removed. Continue degazing for 15 minutes total. The degazing is a convenient time to weigh out 0.1 gram of APS in a small weigh boat and to test its potency as described in the text. Figure 3, degazing acrylamide gel solution. Troubleshooting. Problem: gel solution leaks out of a sandwich during casting. Possible cause: plates and speakers misaligned, rubber gasket not seated properly, glass plates are shaped at edge. Remedy: reline, check that is seated flat, use unchipped plates. Problem: gels fail to polymerize or polymerize incompletely along speakers and sealing surface. Possible cause: solution temperature too low, oxygen concentration too high, insufficient or degraded catalyst. Remedy: bring all refrigerated gel solution to room temperature prior to use. It can help to warm the gel solution to 20 to 25 degrees Celsius after deaeration and allow it to polymerize at or slightly above room temperature. Deaerate gel solution 5 to 10 minutes with at least a water aspirator. Check the ammonium persulfate for freshness. Fresh ammonium persulfate will crackle when water is added. If it doesn't, use a fresh bottle. Make new solution delay. If the polymerization problem persists, increase the ammonium persulfate and the TMED concentration by 50%. Increasing the catalyst concentration is particularly useful when working with low acrylamide concentration. Problem: sample well distorted, causing distorted protein bands. Possible cause: air bubbles trapped under comb. Remedy: remove air bubbles if present. Possible cause: in-reacted acrylamide continued to polymerize after comb was removed. Remedy: rinse wells with electrophoresis buffer immediately after the comb is removed. Problem: die front curves down between brackets fronts at the edge. Possible cause: air bubbles trapped between glass plates at bottom of gel sandwich can be large enough to block current flow and produce a localized frown. Remedy: remove air bubbles if present. Possible cause: gel next to the speakers was not fully polymerized, current leaked down at the edge of the gel, producing a localized frown at edge. Remedy: degaze solution or increase ammonium persulfate and the TMED concentration by 50%. Possible cause: upper buffer chamber sealing gasket pinched closed at center. Remedy: check for proper installation. Troubleshooting. Problem: protein bands fuzzy or poorly resolved. Possible cause: sample volume too high. Remedy: load sample in less volume as a tight layer in a bottom of well. Height of the staking gel beneath the well should be at least 2 times the height of the sample in the well. Possible cause: sample diffused out of the well. Remedy: start electrophoresis immediately after loading sample. Possible cause: gel acrylamide concentration too low. Between brackets, proteins that separate near die front are usually less well resolved than those farther up the gel. Remedy: tailor gel concentration to produce optimal resolution. Use gradient gel to increase sharpness of lower MW bands. Possible cause: sample is degraded. Remedy: keep sample on ice until mixed with SDS. Sample buffer on heat in 70 to 100 degrees Celsius. Water bath for no more than 3 minutes. Two-dimensional gel electrophoresis between brackets 2DE. 2D electrophoresis is a powerful and widely used method for the analysis of complex protein mixture. This technique separates protein according of two independent properties in two discrete steps. The first dimension step, isoelectric focusing between brackets IEF, separates protein according to their isoelectric points between brackets PI. The second dimension step, sodium dodecyl sulfate polyacrylamide gel electrophoresis between brackets SDS page, separates proteins according to their molecular weight between brackets MR relative molecular mass. Each spot on the resulting two-dimensional gel potentially corresponds to a single protein species in the sample. Thousands of different proteins can thus be separated, and information such as the protein pi, the apparent molecular weight, and the amount of each protein can be obtained. A large and growing application of 2D electrophoresis is within the field of proteomics. The, the analysis involves the systematic separation, identification, and quantification of many proteins simultaneously from a single sample. Two-dimensional electrophoresis is used in the field due to its unparalleled ability to separate thousands of proteins simultaneously. The technique is also unique in its ability to detect post and translational modification which cannot be predicted from the genome sequence. Application of 2D electrophoresis include proteome analysis, cell differentiation, detection of disease markers, therapy monitoring, drug discovery, cancer research, purity checks, and microscale protein purification. Isoelectric focusing between brackets IEF. Isoelectric focusing is an electrophoretic method that separates proteins according to their isoelectric point between brackets pi. Proteins are amphoteric molecules that carry either positive, negative, or zero net charge, depending on the pH of their surroundings. The net charge of a protein is the sum of all negative and positive charges of the amino acid side chains and amino and the carboxyl termini. The isoelectric point between brackets pi is the specific pH at which the net charge of the protein is zero. Proteins are positively charged at pH values below their pi and negatively charged at pH values above their pi. If the net charge of a protein is plotted versus the pH of its environment, the resulting curve intersects the x-axis at the isoelectric point. Figure 4. Plot of the net charge of a protein versus the pH of its environment. The presence of a pH gradient is critical to the IEF technique. In a pH gradient and under the influence of an electric field, a protein will move to the position in the gradient where its net charge is zero. A protein with a net positive charge will migrate towards the cassette, becoming progressively less positively charged as it moves through the pH gradient until it reaches its pi. A protein with a net negative charge will migrate toward the anode, becoming less negatively charged until it also reaches zero net charge. If a protein should diffuse away from its pi, it immediately gains a charge and migrates back. It is the focusing effect of IEF which concentrates proteins at their PIS and allows proteins to be separated on the basis of very small charge differences. The resolution is determined by the slope of the pH gradient and the electric field strength. IEF is therefore performed at high voltage between brackets, typically in excess of 1000 volt. When the protein have reached their final position in the pH gradient, there is very little ionic movement in the system, resulting in a very low final current between brackets, typically in the microamp range. IEF of given sample in a given electrophoresis system is generally performed for a constant number of volt hours between brackets volt hour VH concentration, being the integral of the volt applied over the separation time. The original method for the first dimension IEF depended on carrier ampholyte generated pH gradients in cylindrical polyacrylamide gel cast in glass rods or tubes. Carrier ampholyte or small soluble amphoteric molecules with a high buffering capacity near their pi. When a voltage is applied across a carrier ampholyte mixture, the carrier ampholyte with the highest pi between brackets and the most negative charge move toward the anode. Those with the lowest pi between brackets and the most positive charge move toward the cathode. The other carrier ampholyte align themselves between the extreme according to their PIS and buffer their environment to the corresponding PHS. The result is a continuous pH gradient and immobilized pH gradient between brackets IPJ is created by covalently incorporating a gradient of acidic and basic buffering groups between brackets immobilins into a polyacrylamide gels at the time it's cast. Immobilin buffer are a set of well-characterized molecules, each with a single acidic or basic buffering group linked to an acrylamide monomer. Immobilized pH gradient are formed using two solutions, one containing a relatively acidic mixture of acrylamide buffers and the other containing relatively basic buffer mixture. The concentration of the various buffer in the two solutions defines the range and shape of the pH gradient. Both solutions contain acrylamide monomer and the catalyst. During polymerization, the acrylamide portion of the buffer copolymerizes with the acrylamide and beta-acrylamide monomer to form a polyacrylamide gel. Problems that may arise during the preparation and use of gels. Problem: decreased rate of gel polymerization, cause oxygen is present. Remedy: degase the solution. Causes: stock solution between brackets, especially acrylamide and persulfate, are aged. Remedy: renew the stock solution. Problem: formation of a sticky top to the gel, cause penetration of the gel by N-butanol. Remedy: overlay the gel solution with N-butanol without mixing them. Do not leave N-butanol standing on a polymerized gel. Problem: poor sample wells, distorted or broken wells, cause stocking gel resists the removal of the comb. Remedy: remove the comb carefully or use a gel of lower percent T. Problem: wells with a loose webbing of polyacrylamide, cause comb fits loosely. Remedy: replace the comb with a tighter fitting one. Problem: weak staining, cause dye is bound inefficiently. Remedy: use a more concentrated dye solution, a longer staining time, or more sensitive stain. The stain solution should contain organic solvent between brackets E.G. methanol, which strips the SDS from the protein to which the dye may then bind. Problem: uneven staining, cause dye penetration or destaining is uneven. Remedy: agitate the gel during staining and the destaining. Increase the staining and the destaining time. Problem: stain bands decolorized, caused dye has been removed from the protein. Remedy: restain the gel, reduce the destaining time, or use a dye that stains protein indelibly. E.G. Protein Navi MXRB. Problem: gel marked non-specifically by the dye, cause solid dye is present in the staining solution. Remedy: ensure full dissolution of the dye or filter the solution before using it. Problem: contaminants apparent, cause apparatus and or stock solution or contaminated. Remedy: clean or renew them as required. Cause: non-proteinaceous material in the sample between brackets E.G. nucleic acid has been stained. Remedy: try another stain that will not stain the contaminants. Cause: sample have cross-contaminated each other because of the overloading of sideways seepage between the gel layers. Remedy: do not overfill sample wells. Ensure good adherence of the gel layers to each other by through washing of the polymerized gel before application of subsequent layers. Problem: protein bands not sufficiently resolved, cause insufficient electrophoresis. Separating gel's pore size is incorrect. Amount loaded differ greatly. Remedy: prolong the run, alter the percent C and or percentage C of the separation gel. Keep the loading roughly similar in size each time. Problem: small changes in electrophoretic mobilities of a standard protein from time to time, cause constituent of the gel vary in quality from batch to batch or with age. Remedy: use one batch of chemical for as long as possible. Replace age stock solution and reagents. Problem: distortion of bands smeared or streaked, cause protein in the sample are insoluble or remain agitated in the sample solvent. Remedy: use fresh ample solvent and or extra SDS and reducing agent in it between brackets, especially for concentrated sample solutions. Cause insoluble matter or a bubble in the gel has interfered between protein band migration. Remedy: filter the stock solution before use and remove any bubble from the gel mixture. Problem: protein migration uneven between brackets bands bent. Cause: pore size of the gel is unconstituent. Remedy: ensure that the gel solution well mixed and the polymerization is not too rapid. Between brackets, to slow it down, reduce the amount of persulfate added. Cause: part of the gel has been insulated. Remedy: remove any air bubbles adhering to the gel before electrophoresis. Cause: electrical leakage. Remedy: ensure that size speakers are in place. Cause: cooling of the gel is uneven between brackets, allowing one part of the gel to run more quickly than another. Remedy: improve the cooling of the gel or reduce the heat by reducing the voltage or ionic strength of buffer. Cause: band and or its neighbors are overloaded. Remedy: repeat the electrophoresis but with small loadings. Leave gaps between brackets I.E. unloaded sample wells between neighboring heavily loaded sample. If necessarily, modify the gel and or electrophoresis parameters between brackets E.G. See above protein bands not sufficiently resolved, so that the relative mobilities of the protein bands change and they do not interfere with each other. Problem: bands not of uniform thickness. Cause: sample wells was at the very end of the row of wells between brackets, the end will affect. Remedy: avoid using the ends well. Cause: the sample was loaded unevenly. Remedy: check that the sample well bottoms are straight and horizontal between brackets. See above poor sample wells. Bioinformatics. Bioinformatics represent a new area of science that use computational methods to answer biological questions. The complex data sets are used to reach biological conclusions. Types of database: 1. Primary database, raw and redundant data sub. Bioinformatics represent a new area of science that use computational methods to answer biological questions. The complex data sets are used to reach biological conclusions. Types of database: 1. Primary database, raw and redundant data submitted, owned, and updated by experimentalists between brackets E.G. gene bank, SMP, GEO, BubShim, substance, and bioassay. Gene bank is archival, nucleotide-only sequence database. Each record is assigned a stable occasion number. The submission of gene bank data to NCBI is through 1. Direct submission of individual record via web between brackets bank IT sequence. 2. Batch submission of bulk sequence via email between brackets EST, GSS, STS. 3. FTP accounts for sequencing centers. 2. Derivative database, a human created E.G. GEO database, structure and literature databases. 2. Computationally derived E.G. Unigene, homologene, BubShim compound. C. Combination E.G. RefSeq genome assembly, conserved domain, and the structure database. RefSeq sequence database contains the created sequence, standardized nomenclature and record structure, and added a notion between brackets reference sequence futures. It is updated to reflect current sequence data and biology. It is stewarded by NCBI staff and collaborators. The NCBI data model. Why use a data model? Most biologists are familiar with the use of animal models to study human disease. Although a disease that occurs in human may not be found in exactly the same form in animal. Often an animal disease shares enough attributes with a human counterpart to allow data gathered on the animal disease to be used to make inferences about the process in human. Mathematical models describing the forces involved in musculoskeletal motion can be built by imagining that muscles are combination of springs and hydraulic piston and bones or level arms, and oftentimes such models allow meaningful prediction to be made and tested about the obviously much more complex biological system under consideration. The more closely a model follows a real phenomenon, the more useful it is in predicting or understanding the natural phenomenon it is intended to mimic. In the same vein, the National Center of Biotechnology Information between brackets NCBI introduced a new model for sequence related information. The advantages of the model between brackets E.G. the ability to move from the published literature to DNA sequences to the protein they encode, the chromosome maps of the gene, and to the three-dimensional structure of the protein have been apparent for years to biologists using Entrée. NCBI database and visualization tools. One primarily based on nucleic sequence, A gene bank between brackets sequence formatter, B genome between bracket map viewer, C unigene between brackets expression profile viewer, D SMP between bracket gene view and genotype viewer. Two other types of NCBI, A protein between brackets translation and curation seen by sequence formatter, B homologene between brackets protein sequence clusters, C genome project between brackets information for organism sequencing projects, D GEO between brackets arrays and spectra, E structure, F bioassay. NCBI search methods: 1 Entrée, word text search, 2 BLAST sequence search, 3 VAST macromolecule structure search, 4 BUPSHIM structure search, molecular structure search. SEC-IDS. The NCBI data model defines a whole class of objectives called sequence and identifiers between brackets SEQID. NCBI integrates sequence data from many sources that name sequence records in different ways and where the individual names have different meanings. In one simple case, PIR, Swiss Prot, and the nucleotide sequencing databases all use a string called an accession number, all having a similar format. Just saying A10234 is not enough to inequally identify a sequence record from the collection of all these databases. One must distinguish A10234 in Swiss Prot from A10234 in PIR. Because of this, a PDB SEQID contains a molecule name and a chain ID to identify a single unique sequence. Using NCBI data model, there are a number of consequences of using the NCBI data model for building databases and generating reports. 1 gene bank format. In gene bank format, the protein product of a coding region feature is displayed as a translation qualifier, not as a sequence that can have its own features. The gene features name a region on a sequence typically covering anything known to affect the gene's phenotype. 2 FASTA format. FASTA format contains a defined line and sequence characters and may be used as input to a variety of analysis program. The definition line starts with a right angle brackets between bracket greater than and is usually followed by sequence identifier in a parsable form, as in this example, greater than GI 2352912 GB AF0 12433.1 HSDD-T2. The remainder of the definition line, which is usually a title for the sequence, can be generated by software between brackets NUC prot set. 3. BLAST. The basic local alignment search tool is a popular method of ascertaining sequence similarity. The BLAST program takes a query sequence and search it against the entire database of sequences maintained at NCBI. For Entrée, Entrée is an organized system for linking biological data through a text, search, and retrieval engine with virtual workspace of manipulating large data sets. It includes a network of 29 linked databases. Essential components of Entrée database. 1. All molecular database entries are organized by organism, between brackets, taxonomy database. 2. Each record is assigned a UID, between brackets, unique integer identifier for internal tracking. 3. Each record is indexed by data field, between brackets, author, title, organism, and many others. 4. Each record is given a document summary, between brackets, doc sum. 5. Each record is manually or computationally assigned links in biologically related UIDs in and across database. Entrée structure, between brackets, molecular modeling database. Derived from experimentally determined protein database, between brackets, PDB records. Data is added to PDB records, including addition of explicit chemical bonding information, validation and indexing of sequence, inclusion of taxonomy, citation, and other information, conversion to ASN.1 data description language. Searching the structure, database, keyword search by Entrée, sequence search by BLAST, domain search by CDD, RPS, BLAST, structure search by VAST. 5. Sequin. Sequin is a submission tool that takes raw sequence data and another biological information and assembles a record for submission to one for the DDBG, EMBL, GenBank databases. It makes full use of the NCBI data model and takes advantage of redundant information to validate entries. For example, because the user supplies both the nucleotides and protein sequences, Sequin can determine the coding region location. It compares the translation of the coding region to the supplied protein and reports any discrepancy.